Good morning, children. Let's revise the chapter 2, the sexual reproduction in the flowering plants. Now, the flower is the reproductive structure in the flowering plants. It contains the varied structures called the endoecium and gynoecium reproductive organs. The endoecium is the collective term for the stamen, while the gynoecium is the collective term for the pistil, which are the female reproductive structure. This endoecium is present in the form of the whorl of stamen, and this is a male reproductive structure, while the gynoecium, which is a collective term for the pistil or the carpel, it represents the female reproductive structure. There may be one pistil or two pistil or three, so monocarpellary, bicarpellary or tricarpellary. Now we'll study about the stamen, microsporangium and pollen grains. The stamen consists of the two parts: filament, which is a stalk-like structure which holds the anther. The anther is a bilobed structure. Physically it is called the ditheca structure. Two lobes are connected by a soft tissue called the connective. The connective tissue is present. Each lobe or each theca contains two microsporangia. So in all there are the four microsporangia present in one anther, and after maturation each microsporangia develops into pollen sac. The pollen sac contains the pollen grains. Microsporangia is circulant structure, and it contains the four walls: epidermis, outermost, which protect the microsporangium, endothecium, middle layer, and tapetum. Endothecium helps in the dehiscence of the microsporangium, while the middle layers helps in the protection, and the tapetum provides the nutrition to the developing microspores. Young anther contains compactly arranged homogeneous cells. This homogeneous cells is known as the sporogenous tissue. Each cell of the sporogenous tissue is known as microspore mother cell or pollen mother cell or meiocyte. These meiocytes undergo meiosis to produce the microspore tetrad or pollen grain. In a figure of the anther, we'll find the long thread-like structure called the stalk or filament, and in a stamen 2 has two parts: filament and the anther. This anther contains the two lobes. This is one lobe, this is second lobe, so it is bilobed anther. Here this is one lobe containing two microsporangia, and other lobe containing two other microsporangia. These two lobes are connected by this connective tissue. Each microsporangia contains the pollen grains. These microsporangia has a line of dehiscence through which it dehisces or bursts out to release the pollen grain in the niche. This is the cellular structure of the microsporangium. This is the TS of the microsporangia. Outermost layer is the epidermal cell. Then the endothecium. This endothecium has one point where it bursts out. This is called the operculum. The middle layers and the tapetum is utilized during the development of these pollen grains. Initially, the pollen mother cells or myocytes or microspore mother cells are present in the sporogenous tissue. This will undergo the meiosis to produce the microspores. Microsporogenesis. So each microspore mother cell or undergo meiosis, and this meiosis is called the microsporogenesis, to produce the microspore tetrad. Since when a cell undergoes meiosis, it produces the 4 cells. So there is a microspore tetrads are formed. MMC is a diploid cell containing 2n number of chromosomes, while each microspore contains n number or haploid number of chromosomes. So microspore, after maturation, develops into the pollen grains. Each microsporangium produces large number of pollen grains. The size of the pollen grain ranges from 25 to 50 micrometers. It is spherical in structure. The 2 walls are present in the pollen grain. One called the outermost is called the exine and the inner is intine. The exine is made up of a chemical called the sporopollenin. It is highly resistant material. It is not affected by an acid or bases. But there are certain regions where the sporopollenin is not present. These are the regions from where the pollen grains develop. A pollen tube come out. These are the pollen grains of various different species. This is the one pollen microspore tetrad. The 4 pollen grains are present. At maturity, each pollen grain contains 2 cells, vegetative cell and generative cell. Vegetative cell is rich in the carbohydrates, proteins and fat-like materials, which help the generative cell for the division to form the 2 male gametes. Generative cell, since it is small, it floats on the surface of the cytoplasm, as the vegetative cells contains different types of the carbohydrates, minerals. So pollen grains are rich source of the various nutrients. So they are used as the pollen tablets for the human beings. Some of the pollen grains are causes the allergy, like the parthenium or the carrot grass. Pollination generally occurs at the 2 cell stage. So from the anther to the stigma, and when pollen grain reaches the stigma, there is interaction between the stigma and the pollen grain and further development of the pollen grain take place on the surface of the stigma. Thank you.